D4.1 Natural Selection. Our guiding questions are: what processes can cause changes in allele frequencies within a population? And what is the role of reproduction in the process of natural selection? Let's begin by talking about how natural selection is the mechanism that drives evolutionary change. This is the most tested on SL 7-8 mark question and the second most for HL. So it's so important that you can describe natural selection using all of these key vocabulary terms. So let's walk through it. Evolution by the mechanism of natural selection. First principle: organisms produce more offspring than the environment can support. Picture a frog that lays 400 eggs. Some of those are going to be eaten before they even hatch into tadpoles. From there, a lot of those tadpoles are going to be eaten before they can grow into juvenile frogs. From there, many of those frogs are going to be eaten. And at the end of the day, there's only going to be a handful, maybe even only 2 or 3 or 1 or even no adult frogs that end up being able to grow to reproductive age. And so, which one or ones will be the ones that survive? Well, among those offspring, there is variation which arose from random mutations. You need to remember that the source of all variation is going to be random mutations. Now, we have other ways that we can kind of mix up the genes once those random mutations occur, but you need to know that those differences between the offspring are not due to the offspring trying to grow taller or change color, become smaller or faster. All of the differences are due to random mutations. And we'll look at this example in a minute of 2 butterflies of different colors that are within the same species. Okay, the yellow butterfly didn't do anything to make it yellow, and the brown butterfly didn't do anything to make it brown. One of these had a random mutation that turned it to that color. Random mutations occur during DNA replication, and it has to be during DNA replication when an egg or a sperm cell is formed. Now that we have random mutations and variation, some of those variants are better suited to the environment. Therefore, they have a higher chance of surviving to reproductive age and producing offspring, known as having a higher fitness. This is sometimes called survival of the fittest. Now, while we might think in common language of fitness as being like who goes to the gym the most or like who can, bench the highest amount of weight, from a biological perspective, fitness is all about surviving to reproductive age and producing fertile offspring to pass on those genes. So really, in the most simple terms, it's just who can make the most babies that can make the most babies. So whether that's a human, whether that's a butterfly, or whether that's a bacteria. So it's obviously a simplification. If you're talking about bacteria, don't say babies, but hopefully that just makes it clear, okay, that in biology, it's all about producing offspring. Now, those less fit or less well-adapted variants have a higher risk of mortality from predation or other factors, depending on what their random mutation provides for them in terms of survival rate. The features that aid survival are disproportionately inherited by successful offspring. These features therefore increase in frequency in the population. So we can summarize that the heritable features of the population have changed, so it has evolved. Note only features that are heritable, those that can be passed down, okay, are going to make any difference. If, for example, there was some radiation, okay, that caused a mutation in DNA of an adult that led to maybe having skin cancer, that doesn't mean that that same mutation is going to show up in their kids because that happened in a skin cell. The only mutations that are going to pass down are going to be those that occur in the production of egg and sperm cells. These bullet points here, you need to memorize them and you need to know all of that vocabulary. Natural selection then operates continuously over billions of years and has resulted in the biodiversity of life on Earth that we see today. So let's walk through this butterfly example. Here's the same species of butterflies by random mutation. Some are yellow, some are brown. This is an inherited variation. It happened due to random mutation. More offspring are produced than the environment can support. Now, these yellow butterflies make green caterpillars, and these brown ones make brown caterpillars. Note that the brown caterpillars have an advantage because they can camouflage in with these brown trunks that they live on, whereas the green ones cannot. So that overproduction of offspring leads to competition for survival, competition within that species known as intra-specific competition. So then we can see that we have some predators that are visual predators, and they are going to much more easily be able to spot these green caterpillars that don't blend in with their background. Therefore, these brown caterpillars that do blend in have a higher fitness because they have a higher chance of growing to butterflies and passing on those genes to offspring. And you can see over many generations, there's going to be a change in allele frequency. Now remember that alleles are just variations of the gene. And so we're going to have more of those brown alleles in the population just because those individuals have been more successful, in mating and passing on their genes because they've been able to survive with their beneficial adaptation. This is how we define evolution, a change in allele frequency in a population over time. All right, now we've already covered the difference between Lamarck and Darwin. So just as a reminder, Lamarck believed that acquired characteristics during an individual's lifespan could be passed on to their offspring. And so if giraffes just stretched their necks as much as they could, then their babies would be born with longer necks. Now we know this is not the case. Instead, through the mechanism of natural selection, there are going to be some giraffes with variations that are taller and have longer necks than others. Those with longer necks that are more successful in being able to eat enough food can therefore produce more offspring. And over time, we end up with giraffes with longer and longer necks. So in Darwin's time, it was widely understood that species evolved. The mechanism was not clear. Darwin's theory provided a convincing mechanism and replaced the old theory of Lamarckism, but it was a difficult journey for him. There's a lot of great movies that have been made about Darwin's life. If you're interested, you should take a look at them because this was a massive paradigm shift. A paradigm shift is a fundamental change in a framework used to interpret information. And so we saw that when we left Lamarckism behind, people began seeing evidence for Darwinism, that was a massive change in how people viewed the world. It was a paradigm shift. Now, there's a couple of vocab words that you need to be sure that you've mastered. First up, alleles are different forms of a gene. So while we all have the same chromosomes with the same genes on each of those same locations on the same chromosome, we might have different alleles or forms of that gene. And they're still going to be located at the same place, but you get 2 copies of each allele, one from dad, one from mom, and we'll see some different patterns of inheritance that we can conclude based on which alleles are received. You should also know that gametes are going to be sex cells. They're egg and sperm cells that contain half the genetic information of the parent. They're haploid. Now, while random mutation is where all variation begins, it produces all of those new alleles during mistakes made during DNA replication of sex cells, so in the production of those gametes. These random mutations can be harmful, they can be beneficial, or they can have no effect. Once that variation exists by random mutation, there's 2 other ways that those genes can then get sort of mixed up and recombined. The first one is meiosis. This is the process of cell division that produces those gametes. And so it halves the individual's genetic information, but it halves it differently every single time, which is why we have different eggs and different sperm within the same individual. This is why brothers and sisters look different from each other. If every single egg produced by a female was the exact same genes and every sperm was the same, then every single offspring of the same parents would have the same exact genome, like identical twins. That is not the case because every egg and every sperm are different thanks to the process of meiosis. Next, a different way that also provides that variation once those random mutations have occurred is just sexual reproduction. Which male and which female gamete end up fusing? We're going to have new combination of alleles from two individuals. And so if there is mutations that occurred in different individuals, at this point they can be brought together. While sexual reproduction involves two different individuals, each having their genes and then recombining to form a different offspring, asexual reproduction is when an organism basically clones itself. And so its offspring is an exact genetic copy of itself. And so the only source of variation is going to be that random mutation that might occur. Now, the problem with asexual reproduction is if you're in a changing environment, if every organism in a population is genetically identical, then it may not be able to evolve quickly enough to respond to environmental change. For example, if you have a population of Amazon mollyfish that all have identical blue tails, and a new predator is introduced that targets fish with blue tails, then every single one of those fish are going to be targeted and eaten. Whereas if there was some genetic variation and there was some color variations in the tails and some were blue and some were green and some were yellow, right, there might be a chance that those other colors might survive. And so when we're looking at things like new diseases that show up, the hope is that through random mutation, some members of the population are going to have resistance thanks to random mutation. And so they'll be able to respond by some members being able to survive the new disease and pass on the genes for resistance to the next generation. Next step, carrying capacity is defined as the size of a population that can be supported by the environment. It's indicated here by this red dashed line. You can see here the population size is growing slowly, experiences exponential growth, but then that growth begins to slow as carrying capacity gets nearer, and then eventually plateau. And this is the point where the environment can support the population size. It's determined by some type of limiting resource that's in the shortest supply. This could be things like food availability for animals or light availability for plants. So as Darwin pointed out, more offspring are produced than can be supported by the available resources, leading to a struggle for existence within a population. Not every individual will be able to obtain enough to allow them to survive and reproduce. Next, abiotic factors as selection pressures. So remember that we're studying biology. Ology means the study of, and bio means life. And so bio means life, biotic factors are living factors such as food, mates, predation, competition with other organisms for resources. And so therefore abiotic factors are those non-living factors. This could be things like available space, temperature, altitude, or amount of sunlight available. Let's compare now some density-dependent limiting factors to density-independent limiting factors. The definition of density-dependent limiting factors is that as the population density increases, the limiting factor becomes stronger. As you can see here, there's lots of available nesting sites if it's a small population. However, if we have a large population size, those nesting sites are going to be very, very difficult to find. And so there's competition for those available nesting sites. And so this nesting site availability is an example of a density-dependent limiting factor. These factors are going to tend to be biotic or based on living things. As population density increases, competition for food becomes more intense is another example. Other examples include predation, disease, parasites, and waste accumulation. Now, in contrast, density-independent limiting factors is when the size of the population has no effect on the population density. These tend to be abiotic. For example, if there's a period of freezing weather, it might kill some plants but not others, but the chance of that individual plant being killed by the freezing temperatures is the same whether the population density is high or low. The same thing goes for heat waves. Or just picture any sort of natural disaster. Say there's a plant that lives on a hillside and it only can handle fresh water, and there's maybe a tsunami that washes up on that hillside, and now all of that entire hillside, okay, is now salty soil, and it's going to kill everything because of the osmotic issues those plants experience. Now, if there was one plant on that hillside or 1,000 plants on that hillside, they all die from the salty soil. And so it's density independent. And these, again, are usually abiotic factors, non-living factors. There's that salinity example I just gave you. Earthquakes, fire, pollution, extreme climate conditions such as heat waves or drought are all examples of density independent limiting factors. Remember that we define fitness as whether or not an individual survives for long enough to be able to reproduce, and of those that survive, how many offspring they have. So you can see here, this green beetle has a lower fitness. It only had 2 offspring, 4 offspring in the following generation. Whereas you can see here, we define this beetle as having higher fitness because it had more offspring down the generations. A common misconception about evolution is that adaptations acquired during the lifetime of an organism can be inherited by offspring. Remember, that was Lamarck's incorrect idea. Another example of that would be, say someone is a really great tennis player and they play tennis with their right hand and their right arm gets super strong and agile. Does that mean that their baby is going to be born with a strong right arm? No, of course not. That's preposterous, right? We know that the baby would have to practice and do all of that work to get a strong right arm as well. Those things that are acquired during the lifetime don't get passed on. It's important to understand that those characteristics acquired during an individual's life due to environmental factors are not encoded on the base sequence of genes, and so they're not heritable. Adaptation doesn't involve trying, right? This little beaver can't just think, chainsaw, 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 and all of a sudden grow a chainsaw in order to cut this tree down. We had already talked about how this giraffe can't stretch and stretch its neck and be able to pass on a longer neck to offspring. Acquired traits during an individual's lifetime are not passed on to offspring. A heritable mutation is a change to the base sequence of DNA due to a mistake during DNA replication that occurred during that DNA replication process during the production of an egg or sperm cell that, after fertilization, became the new offspring. Next, sexual selection as a selection pressure in animal species. Now, half of the genes in an animal's offspring comes from its mate. So there's going to be an advantage to assessing accurately whether a potential mate is well adapted to its environment in order to ensure the greatest chance of survival for the offspring. So if you are trying to invest in having the strongest offspring, they're going to be able to keep passing on your genes, you're going to want to choose a mate that is strong and healthy. And so we're going to see sexual selection come into play. Differences in physical and behavioral traits that can be used as signs of overall fitness can affect success in attracting a mate and so drive the evolution of an animal population. Courtship behavior includes the ability of a male to overpower other males by fighting. If the female sees that this male, say, is stronger and can fight off of this male, then this might be the male that she chooses because then she knows her offspring would have the best chance of winning in a fight. Anatomical features that appear to the human eye to be excessive, like the tail feathers of a male peacock. However, if that peacock can get enough food and it can avoid potential predators while still having this extremely large tail, then that means that peacock is probably going to have good, healthy genes to pass on to potential offspring. And then behavior patterns that seem bizarre, like the plumage and courtship displays of a male bird of paradise. The birds of paradise is a specific example that you should be familiar with. There are 40 species of birds of paradise living on New Guinea and nearby islands. The males have very showy plumage with elaborate feathers that have no use in flying. The females, which build the nest, are relatively drab. Now, the males of many species have a complicated courtship dance that they use to try to attract females. The colored plumage and courtship dances help to avoid interspecific hybridization by allowing females to determine if a male belongs to their species. Now remember, if a horse and a donkey were bred, they would produce a mule. So while they would be able to produce offspring, mules are infertile because they end up with an odd number of chromosomes. And so that's a waste in terms of fitness. If you can make offspring, but the offspring are not passing on your genes further, that is not a good choice from a biological perspective to have a high fitness. And so by having specific courtship dances, the females can recognize that that individual is definitely of the same species and therefore would have the highest chance of producing fertile offspring. So these crazy bright plumage, they're super long, not used for flying. They make these birds very visible to predators by being bright colors. Darwin explained such traits in terms of mate selection. Females prefer to mate with males that have exaggerated traits. The reason may be that these traits indicate an overall health. For example, if a male has enough energy to grow and maintain the elaborate plumage and repeatedly carry out vigorous courtship displays, it indicates the male must have fed efficiently. And if a male can survive in the rainforest with the encumbrance of all their tail feathers, with bright plumage that makes it highly visible to predators, it's probably well adapted in other ways. And therefore, that male bird is going to be a good mate to choose. Males with showier plumage and more spectacular courtship dances then produce more offspring and continue the traits, and they get passed on from generation to generation. Natural selection has therefore caused these traits to become exaggerated. A specific example of sexual selection is the work of John Endler and his colleagues. They studied guppies in Trinidad and Tobago, and they looked at the effect of predation on the guppy's physical traits. Now, in the end, Endler found that coloration gives an advantage to males, as females prefer colorful males for mating. However, it can be a disadvantage in shallow water as it makes the guppies more visible to predatory fish. And so he concluded that in the parts of the stream where there were fewer predators, the males were more colorful, right? It was less selected against. Males in locations where there were more predators tended to be less colorful. And as an application of skills, you're expected to interpret data from John Endler's experiments with guppies. Here I have some background from the practice DBQ in the textbook, where we had just guppies in pond Q, guppies with a weak predator in pond R, a strong predator in pond S. I recommend pausing this, reading the background, and then taking a look at the data. Make sure that you can interpret it. I have the answers listed for each of the questions. So again, I recommend that you're going through this slowly, carefully, pausing along the way, making sure that you get that practice DBQ in and that you're familiar with John Endler's work. All right, this takes us to the end of SL content. So SL students, you can go ahead and stop the video now. HL students, stick around, we have more to go. So next up, the concept of the gene pool. First up, you should be able to define a population. A population is a group of organisms of the same species living in the same area. A gene pool is then all of the genes and their different alleles that are present in a population. Note, all of these warthogs that live in one particular area of the savanna are considered a population. They're living in the same place at the same time, and they're the same species. So all the different forms of their different alleles make up the gene pool. It's not a literal pool. It just means all of the different genes that are present and their different forms. So the individuals that reproduce contribute to the gene pool of the next generation. Therefore, fitness can be measured in terms of contribution to the gene pool. The more genes that are added to the gene pool, that's because you've got more offspring that are being produced. Species that consist of multiple populations may be reproductively isolated from each other due to geographical separation. Now, in this case, individuals can potentially interbreed with each other, but don't actually do so. The separated populations have their own gene pools. It is therefore possible for multiple gene pools to exist in one species. Okay, this is a map that is showing human populations and a particular allele frequency shift between different geographical areas. So you'll see that in humans, there are differences in allele frequencies between ethnic groups, different geographical regions. So as an application of skills, students should use databases to search allele frequencies. Okay, there are many different allele frequency websites out there. One example is Alfred, very cleverly the allele frequency database, which spells out, if you take those first few letters, Alfred. Okay, there's also other ones. If you don't find that one particularly user-friendly, just do a Google search, but you should take some time and just browse an allele frequency database and take a look around and see what you see. Okay, now we have talked about natural selection. We went through that first slide with all of those critical steps. Overproduction of offspring. Variation comes from random mutation. And we know that those organisms with the highest fitness are going to be able to pass on their genes, and we're going to then have more of those particular variants of the gene in the next generation. Now, think back. Darwin published his theory in 1859. That's when he published On the Origin of Species. It wasn't until almost 100 years later that they even figured out the structure of DNA. Darwin did not understand genetics at all like we understand it today. And so we have now what we call Neo-Darwinism, where we explain Darwin's same ideas, but we're going to integrate our new genetic knowledge into Darwin's ideas. And so instead of just what I said earlier about natural selection, we can now take it one step further and describe that same process by saying that evolution is a change in the frequency of alleles in the gene pool. So the alleles are forms of the gene, and we have more of those alleles in the gene pool, okay, for those particular variants that are more successful, that have higher fitness. If an individual is better adapted to its environment than its competitors, it will tend to have more offspring. Therefore, its genotype will increase in frequency in the next generation relative to other genotypes. Make sure you can use this vocabulary when discussing natural selection, be able to connect it with what we know about genetics. Next up, there are 3 patterns of natural selection: directional selection, stabilizing selection, and disruptive selection. All 3 types result in a change in allele frequency. First up, directional selection. This happens when the selection pressure is acting upon one side of a spectrum of traits. The population is going to change as one extreme of a range of variation is better adapted. For example, bird beak size. Our example of the medium ground finch. Okay, the selective pressure is that during dry years, small seeds are not abundant, and so birds with larger, tougher beaks become more frequent. Now, the common error here is for people to say, or even just write in a way that assumes that the birds are somehow growing bigger beaks, like they're trying to get a bigger beak because there's only big seeds out there because of a dry year. That is not the case. These birds are not growing bigger beaks. Go back to step 1. Because of random mutations, there is variation in bird beak size. Some birds are going to have smaller beaks. Some birds are going to have larger beaks. And it's the ones that have larger beaks by random mutation that will survive longer and be able to pass on those genes so that we see a shift in allele frequencies as the generations progress over time. So just be very careful that you're avoiding anything that could even sort of sound like Lamarckism. Make sure you're discussing this in terms of natural selection and Darwinism. Next up, stabilizing selection is when the selective pressure is acting on both of the extremes, and so it removes extreme varieties. Our great example of this is human birth weight, because if the baby is born at too small of a birth weight, okay, it could lose heat too quickly, it could get ill from infectious diseases more easily. On the other end of the spectrum, babies born with a high birth weight are going to be much more difficult to deliver. And so we have stabilizing selection. Okay, where you can see here the original in orange moves towards the center here in red. Okay, we're going to see that we have selective pressures on both extremes. We're going to end up with more medium weight babies. They have a much lower mortality, and hence the frequency of medium weight babies increases. Next step, disruptive selection. Now this is the opposite. This is where the selective pressure removes those intermediate varieties, instead favoring the extremes. Now, example of this is coho salmon. Coho salmon do external fertilization. Okay, when the female spots the male nearby that she wants to mate with, she'll lay the eggs, okay, and the male salmon will fertilize them. Now, there's two distinct size males that are favored. The small sized males are specialized at sneaking in and fertilizing those eggs, and the large sized males are specialized at fighting off smaller males. And so those intermediate sized males don't have an advantage, right? They can't fight and they can't sneak. And so they are going to have less of a chance of fertilizing those eggs and thus passing on their intermediate sized genes. And so we're seeing disruptive selection, and this example could eventually lead to speciation, where we would have a larger variety of coho salmon, okay, that could potentially eventually no longer interbreed with a smaller variety. Disruptive selection favors the extremes by selective pressures acting on the intermediate varieties. All right, here is a summary of the three. It's a good idea to pause the video here and try to make sure that you can explain each of these along with an example. Next, Hardy-Weinberg equation and calculations of allele or genotype frequencies. Before we go on, it's important that we do a quick genetics review. Now, this might be from an earlier biology class or all the way back to middle school. Okay, so dig deep. If you don't know this, it's time to learn it now. First step, we already established that different versions of genes are called alleles. Individuals have two alleles from a gene, one from mom and one from dad. Alleles have a dominant and a recessive form. If an individual has one dominant allele and one recessive allele, it's the dominant allele that will be the one that is displayed in the individual's phenotype. The phenotype, just like the word physical starts with PH, okay, is going to be the physical observable trait. The dominant allele we indicate with a capital letter. Usually we use the first letter of whatever the dominant trait is. And then the recessive allele, we indicate it with the same letter, but lowercase. For example, big F could indicate the allele for freckles. If it's dominant, it would be capital, and we would use F for freckles because it's the dominant allele's first letter. Little f would be the allele for no freckles, which is the recessive trait. Now, if an individual got a big F, meaning big F from one parent and big F from the other, we would say that that is a homozygous dominant individual. Homo means the same, and dominant refers to that capital F, the gene for freckles. And so if you have big F, the phenotype is freckles. Now, if you get one big F from one parent and a little f from the other, we call that heterozygous. Hetero is a prefix that means different. In this case, because big F is dominant over little f, the phenotype is still freckles. In fact, from the outside, big F and big F little f are identical. There's no way to tell which of these two genotypes an individual has because they both have the same phenotype. On the other hand, if we have little f, we call that homozygous because that's still the same, but now it's homozygous recessive because there's two little f's. And in this case, the phenotype then is no freckles. So the Hardy-Weinberg equation can be used to predict allele frequencies from genotypic frequencies. It can also be used to predict genotypic frequencies and allele frequencies from the incidence of recessive phenotypes. And we'll look at two problems where we do that. But first, here's what you need to know. Little p represents the frequency of the dominant allele. It's the same as, say, big A. Q represents the frequency of the recessive allele. It's the same as little a. P plus Q is always going to equal 1. So the frequency of the dominant allele plus the frequency of the recessive allele equals 1. Now those are single alleles. So now let's look at the genotype where you've got 2 alleles, one from each parent. Now if you had big A, remember that big A is P, and so if you have 2 of them, we call that P squared. P times P is the frequency of individuals who are homozygous dominant. Now, if you have PQ or QP, those are both ways that we can get the heterozygous form. And so we have 2PQ representing the frequency of individuals who are heterozygous. And then Q square is the frequency of individuals who are homozygous recessive. And so we can have our Hardy-Weinberg equation now by saying that our P squared plus our 2PQ plus our Q squared equals 1. You can be given a frequency of a trait in a population. Most of the time, you're going to be given a Q squared. And if you're given a P squared plus 2PQ, you can subtract that from 1 to get your Q squared. Okay, but you're not going to be just given P squared or 2PQ because remember, both of these have the same phenotype and we can't tell them apart. And so we always are going to have to start with Q squared, whether it's given to us directly, like in these next 2 problems, or you will see problems where you're given what dominant phenotype's frequency is, but that is just your P squared plus 2PQ together. So let's look at a practice problem. Albinism is a lack of pigmentation in skin and hair caused by a recessive allele. Albinism occurs in North America in approximately 1 in 20,000 people. Let Q represent the frequency of the albinism allele and P represent the frequency of the pigmented allele. To exhibit the albino phenotype, a person must be homozygous recessive. The probability of having 2 alleles for albinism is Q squared. What is the probability of the heterozygous phenotype? So the first thing that you want to do is just figure out what P and Q are. Because once you figure out P and Q, you can answer any question here. In this case, they're asking the probability of the heterozygous phenotype, but they could ask, what is the probability of the homozygous dominant phenotype, right? Or the homozygous recessive, although that's given. Okay, and so it's really important that we just solve for P and Q. And remember, they're almost always going to give us our Q squared, okay, our frequency of our recessive allele. In this case, it's given as 1 in 20,000 people. So we're going to begin by stating that our Q squared is 1 in 20,000. So we can then solve for Q by taking the square root of both sides. Using a calculator, I found out that Q is 0.007. Next step, once you have Q, you can solve for P because remember, P plus Q equals 1. So if I plug in the Q I just solved for, I just subtract that from 1 to get my P. Now, once I have P and Q, I can give anything that the question asks. So again, we're asked for the probability of the heterozygous phenotype. And so we know that's going to be our 2PQ. So I just take my 2, I multiply it by my P that I solve for here, I multiply it by the Q that I solve for here, okay, and I get my answer of 0.0139. Let's do another example. Example 2. Tay-Sachs disease is an autosomal recessive disorder of the enzyme hexosaminidase. The disorder causes a buildup of fatty deposits in the brain. A child affected by the disease usually dies by the age of 4. The frequency of Tay-Sachs disease, genotype t, in a Mediterranean population is 0.0003. Calculate the frequencies of 2 things, allele t and genotype T. So remember, you're usually given Q squared, and sure enough, you were given the frequency of the disease, which is our Q squared. So if Q squared is 0.0003, the first thing I'm going to do is I'm going to square root that so I can get my Q, which is 0.017. Now I know P plus Q equals 1, so I can subtract that Q value from 1 to get my P. Now once I have Q and P, I can do anything with it. And in this case, we were asked for the frequency of allele little t, which we know is going to be the same as Q. Q is our frequency of our recessive allele. So that went here. And then our frequency, and again, we're asked here for the heterozygous genotype. So that's our 2pq, which we can just plug in our p and q and solve for that answer. Now, what's important to know is that the Hardy-Weinberg frequencies are only expected if the following series of conditions is met in a population. These 5 conditions need to be memorized. Number 1, there is no mutation. Number 2, mating is random. Number 3, there is no immigration or emigration. Number 4, the population is very large. And number 5, there is no natural selection. If those genotypic frequencies in a population fit the Hardy-Weinberg equation, we can conclude that all 5 of these conditions are being met. If, though, our frequencies don't fit what we predicted using the Hardy-Weinberg equation, this indicates that one or more of these conditions are not being met. Perhaps mating is non-random, maybe survival rates vary between genotypes. Okay, if we can be sure that the first 4 conditions are being met, then natural selection must be favoring one phenotype over another, causing allele frequencies to change, and therefore we can determine that evolution is occurring within the population. This is a review. We've already talked about how artificial selection is carried out in crop plants and domesticated animals by choosing individuals for breeding that have desirable traits. Okay, and humans have been artificially selecting species for thousands of years. But now we can tie this in with our understanding of natural selection. Here is what's arguably the point that will show up on every single mark scheme. If there's ever a question about natural selection, make sure that you state there is variation in a population due to random mutation. Then humans repeatedly select for and breed the individuals with the traits most suited to human uses. Here's that wild mustard plant. Okay, and remember that humans selected for different traits, right? They chose the individual plants that had the largest flower clusters, and over thousands of years, we eventually ended up with cauliflower with these large flower clusters. Okay, we also looked at doing this with domesticated animals like cattle that were bred for either meat or milk. Okay, we looked at pug evolution, where pugs were selected for their size and their length of their nose. Okay, and so over thousands of years, or in this case, over a much shorter time, right, just over 100 years, we can actually see a huge change in that population based on humans choosing traits. Other examples of this are different dog breeds that originally came from the wolf. You can see here we've been selecting for larger and larger chickens just over the past 50 or so years. We can see differences in wheat and corn. We can see these wild-looking pigeons that have been bred as fancy pigeons. Okay, and then here's that wild mustard that was on the previous slide, and all of the different foods that we have created by breeding for specific traits. Now, one important thing to note is that artificial selection is done by humans with intentionality. And unintended consequences of human actions, such as the evolution of resistance in bacteria when an antibiotic is used, okay, is due to natural selection rather than artificial selection. Now, while this is, of course, human caused because we are using these antibiotics oftentimes wrong or overuse, but that is still an example of natural selection. Remember that artificial selection has to be humans choosing organisms to breed for desired traits. All right, now that takes us to the end of D4.1. I have here, if you want to pause this and go through this study guide, I have 2 pages for the SL content. Note that this is directly from the IB published guide. And so it's a good idea to just pause, read through this left column, okay, and then read through everything on the right that has sort of specifics about what IB would like you to know. All right, so there's slide 1, and then just a little bit more here on slide 2. And then lastly, I have a slide here for just the HL content. All right, folks, good luck out there.